Hello friends, welcome back to my channel Medicine of Manzuran. Today we'll talk about the physiology of the thyroid gland and its clinical correlates also. Okay. Now see, when you are talking in terms of the thyroid gland, basically we know what is the thyroid gland is the butterfly shape, the endocrine gland that is present in front of the trachea and the neck regions. Okay. So if we see the inner structures, we'll get the two portions for this ones, and that will get what by the isthmus. This is the mid portion, that's the isthmus that will connect the two major portions of the thyroid gland. Okay. Now, within the thyroid gland, what type of the structures we'll see? We'll get the multiple, this type of the structure, they are known as the follicles. We'll get the multiple, this type of the follicles. Okay. And each follicles is having all the thyrocyte cells. The follicles are made up of what? Thyrocyte cells. So, for example, if I say, these are thyrocyte cells, these are thyrocyte cells, and whenever we'll get the combination of the thyrocyte cells, they will form the follicles. And within the thyroid glands, we'll get the multiple follicles. Okay. Now, these are the known as also the follicular cells over here. Okay. Now see, this is the blood vessels through which the blood that will be supplied from here to the thyroid gland, and then we can say in terms of also in terms of thyroid follicles also. Now see, around these structures we are getting there are some clusters of some cells that are known as parafollicular cells. They are known as parafollicular cells. This is there also the parafollicular cells. Now if we talk in terms of that, try to understand from here, as we know that these follicular cells, cells, they are responsible for productions of the T3 and T4. I'll talk in details all of these things, how the T3 and T4s are formed. And to talk in terms of the what, the parafollicular cells, they are responsible for secretion of what? Calcitonin hormone, right? The calcium lowering hormones. So the parafollicular cells. Okay. Parafollicular cells, they are responsible for secretion of what? Calcitonin. So calcitonin that will be secreted by the parafollicular cells that is present near the follicular cells, and they will basically what's the main functions? Their main functions for lowering the calcium level. Now, if we say in terms of the normal functions for the thyroid gland, what's the main function for the thyroid gland or the thyroid hormone over here? If we say the functions, as far as the function is concerned, we can say the main functions over here. First, the mental growth. The mental growth. We can say also in terms of what, the physical growth. And also what's the BMR, they maintain the basal metabolic rate. Another functions for them, you can remember from here that they increase the sensitivity. To the adrenergic receptors. So increase sensitivity. You can write it over here. Increase sensitivity to adrenergic receptors. So adrenergic drives. That's the main functions for them. Okay. Now we'll go through in details about how the hormones are formed. Now, if you want to make the thyroid hormones, if you want to make the T3 and T4, the raw materials would be the what? We need to have the raw materials. And what is the raw material? There is the iodide. So the iodide that should be taken from what? From the GIT, from the diet. So from the diet we'll get what? First things, what are the requirements to produce the T3 and T4? We can say over here the thyroid hormone. First we have to get what? The iodide. So first we will get the iodide. And whenever we get the iodide, after that whatever the thing we need to have, okay, let's write one by one the requirement things. Okay. See, I'll explain all these things. First, let's write what are the requirements to produce the T3 and T4. First things, we have to have what? The raw material. That is the iodide. The second thing, I'll talk in details. Wait. We need to have what? The thyroglobulin protein. Okay. Thyroglobulin protein. That will be formed where? Within the thyrocytes. It will be formed within the thyrocytes. These are the thyrocytes. I'll talk about all these things. Okay, don't worry. So where? Within the thyrocytes. Then we need to have, along with that, okay, let's use this one. Then we need to have, whenever the iodide we are getting over here, from here the iodide it will go within the circulations, and from the circulations we will get the iodide within the follicular cells. within the follicular cells. And these cells, they will also produce what, the thyroglobulin protein. They will also produce thyroglobulin proteins. After that, these things that should be released from here to the within the lumen. So we can write it over here that releasing of, you know, iodide molecule and what, thyroglobulin protein, thyroglobulin protein wire within the luminal site. And these things, whenever try to understand is thyroglobulin cells, that means if we say is thyrocytes within the follicular structures, they will secrete what from here. Try to understand, they will take the, you know, iodide from the circulations and they will just release the iodide over here. It will also release the iodide over here, release the iodide. It will also release the iodide. It will also release the iodide. In the same way, all the thyrocyte cells will what, release the iodide molecule. Okay. Along with the iodide, they will also release what, the thyroglobulin proteins. So we get also the thyroglobulin proteins that will come from the each what, thyrocyte cell. And whenever we will get these collections of, try to understand, whenever we will get the collections of the thyroglobulin proteins along with that what, iodide molecules, that will be taken from each individual what, thyrocyte cells. Now we are getting the depositions or accumulations of the iodide molecules and what, the thyroglobulin protein known as colloid. So this whole figure, that is known as colloid. Okay. Colloid. You get the colloid formations. Okay, then we'll get the colloid formations and storage of them. Storage of the what? Iodide and thyroglobulin protein over here. Okay, and after that, whenever within the circulations we need from here, we will get the productions of the T3 and T4. Okay, I'll explain in detail. So now see, whenever we already made the T3 and T4 over here, whenever we need within the circulations the T3 and T4 for using within the different kinds of the tissues, at that time this T3 and T4, they should come from where? From the colloid to the, they will back from colloid to the, you know, thyrocyte cells. From here they will come within the circulations within T3 and T4. Okay, now we will talk about all the mechanisms, how they are getting formed and what type of the enzymes, you know, what type of the, you know, pathways are involved over here. Okay, so let's draw this whole part, then it will be more clear for you. Okay, so for that thing I need to erase this whole part. I am erasing this whole part. Okay, now we'll try to understand from here. I'll just take a single from here. I'll just take a thyrocyte cells and we'll see what type of mechanisms are involved to produce the T3 and T4 hormones over here. So for that thing, okay, I'm just taking in this way. Okay, let's take from earlier part, and I think it will be easier for me and easier for you also to understand the whole part. We get one over here. In the same way, we'll get the thyrocyte cells from here also. In the same way, we'll get also the what? The thyrocyte cells from here also. We'll also get the, okay, let's erase this part also. We'll also get the thyrocyte cells from these all directions. For example, from here. Okay. Now, at first try to understand from here, these all are the what? Thyrocyte cells. That means these, the follicular cells over here. Now, within these structures, these are the known as what? These are the known as basal sites. These are known as what? Basal site of the cells. These are the basal sites of the cells. This is the apical site, you can say. And definitely it is the luminal site. It is the what? Luminal site. It's a luminal site. Now, try to understand. We have already said we have to get what? We have to get the iodide in terms of the raw material from the GIT. So, for examples, we have taken the food from where we have. Okay, let's do from here. Now, from here we already got the what? Iodide. Now, this iodide that will come where? Within the circulation. Within the circulation we will get the what? Iodide. So, whenever we got the iodide within the circulations, from here The iodide is supposed to go where? Okay, let's use the different color. We got the iodide within the circulations from the GIT. Now, this iodide, try to understand this iodide, it's a charged molecules, and as we know that the membrane is not permeable for the charged molecules, the polar molecules. Okay. Another thing, see, the concentration of the iodide is usually more with respect to outside of the cells. And another thing, try to understand from here. If we say under normal circumstances, the cells would have the resting membrane potential that is negative, and the iodide is negatively charged, so it is already negative, and we are just what? We are just forcing the iodide molecules to take within the cells. So definitely here we are getting two things. We are just pushing the iodide molecules against the concentration gradient of the iodine along with the what? Electrical gradient of the iodine. That's why we need to use some active transport mechanisms to take the iodide within the thyroid cells. So that's why we get over here the iodide pump. This is known as what? Iodide pump. Why iodide pump? It is active mechanisms because we are just taking iodide molecules against the concentration gradient, along with the what? Also the electrical gradient. Now see, whenever you are just sending the, okay, let's use this color only. We have already got the, by using the what? Active transport mechanism by the iodide pump, we have got already the what? Iodide. Now we have got already the iodide. We got the iodide. Now whatever thing we'll see further. So whenever we got the iodide over here, this iodide that will be taken within the thyroid cells. Now we have to send the, I have already said the mechanism, in the steps that iodide should be taken within the colloid parts, that means within the luminal part, within the lumen. Now here is present on top of the enzymes, where? Within the lumen. Here we will get on top of the enzymes. This enzyme, they will do what? They will just convert the iodide molecules into iodine molecules. They will convert the iodide molecules into what? Iodine molecules. Okay, perfect clear. Now what is the name of this enzyme? The name of this enzyme, that is known as thyroid peroxidase. So if you write the name of the enzymes over here, so the name of the enzyme is over here, we can write thyroid peroxidase. So we'll get the oxidations over here, and by the process of oxidations we'll see the negative charge that will be removed and the iodide molecule that will be converted into normal iodine. The enzyme is having the many functions. So first functions I just described over here. They will just what? They will convert the iodide into iodine. The next functions over here. This enzyme will give the many signals. One of the signals that will come within the thyroid cells. And within the thyroid cells, what do we know? There is a presence of the nucleus. And we will get within the nucleus whenever the signals will come. Certain genes, they will be activated, and they will produce the messenger RNA. And the process of the protein formations. First we will get the mRNA, and these activations of the genes that will be done by the what? Thyroid peroxidase enzyme. Okay, so we'll get the mRNA that will be translated, and basically we'll get what? In the translations we'll get that they will go where? Within the rough endoplasmic reticulum. After that they will go where? The processing will be done where? Ultimately where? Within the Golgi apparatus. Actually the total processing will get what? The packing, all these things that will be done where? Within the Golgi apparatus. Ultimately we'll get the protein. Okay, I'm just showing with the dot color. It will go through the rough endoplasmic reticulum. Then we will go Golgi apparatus. If you want to write, you can write. Okay, then we will get the proteins. For example, I'm showing the protein with this color only. Okay, this protein. Now, what is the name of this protein? That is known as thyroglobulin. In the video, I'll mention the thyroglobulin in terms of T.G. That means try to understand this stands for the thyroglobulin. Now, thyroglobulin basically when you are talking in terms of the protein, definitely it will have about the amino acids. Now, in this cases, on amino acids, I need to highlight over here the tyrosine. We'll get the many amino acids, but for example, we are getting over here the tyrosine. Now, along with the tyrosines, we'll get other amino acids also. For example, we are getting the others amino acids, but I am highlighting over here the tyrosine. This is the tyrosine. Why did I highlight? You will come to know right now only. After that, what type of functions we will see? This enzyme, the thyroid peroxidase, they will take these molecules. That means what? The thyroglobulin proteins within the lumen. Now whenever we will get this thyroglobulin protein were within the lumen, for example, we got the protein over here. Okay, we got the protein over here, and within the proteins we got basically, or specifically, we'll get what? The tyrosine molecules. We'll get the tyrosine molecules over here. Okay, now we have already that the tyrosine molecules along with the thyroglobulin proteins over here. Now what's the next function for the thyroid peroxidase enzyme? Now they will do what? This enzyme will add this iodine molecule, whatever the iodine molecule we got over here within the lumen, they will add these iodine molecules within the tyrosine molecules. So they will add the iodine molecules within the tyrosine amino acids. So first try to understand. So we can see that the iodine molecules. So whenever we will get the additions of the iodine molecules over here, on iodine molecules can be added over here. We can get over here sometimes the two iodine molecules that can be added in one. Sometimes on we can get 2, we can get over here 2, we can get 1, we can get 2. Okay, now see, whenever we are getting, try to understand from here, I am showing this thing over here. Whenever we are getting, try to understand from this figure. See, for example, is the tyrosine molecules, is the tyrosine molecule, see. Whenever we are talking in terms of the on iodide, for example, on iodide that is getting added within the tyrosine molecule. Now this thing is known as monoiodotyrosine. If I say this thing that the two iodide molecules are getting connected with the tyrosine molecules, then this thing is known as diiodotyrosine. For example, over here 1 over 2, then it will be known as what? Monoiodotyrosine. It will be known as diiodotyrosines. 1, 2, whatever you can take it. The same way, we'll get all these things in the whole protein parts. Okay. Now what's the next thing? So we are getting over here the additions. We got what? Okay, if you write down the functions of the enzymes, so let's write it over here. What's the functions? First functions we got over here. The iodide, that means the iodide that is converted into iodine. The second thing we got the thyroglobulin formation. Third thing we got over here. They are just adding the what? They are adding the iodine molecules along with the what? They are adding molecules. That means this enzyme is helping to attach the iodine molecule along with the tyrosine. This thing is known as organification. Now try to understand, whenever this iodide, that will be added what? With the tyrosine molecules. This thing is known as organification. After that, whatever thing will get over here, try to understand. Okay, so you already got over here. Then this enzyme, that means this thyroid peroxidase enzyme, they will just do the fusions in between the tyrosine molecules. They will just do the fusions. For example, they will just fuse these two tyrosine molecules along with this. They can fuse this one, they can also fuse this one also. Okay, and for example here one more tyrosine molecules I am taking, they are fusing this one over here. And this thyroid peroxidase enzyme, what they are doing, they are fusing the two subsequent what? Tyrosine molecule with each other. So whenever they are fusing with each other, whatever the structures we are getting over here, try to understand. Here the 2 and 1. So total how many iodine molecules are getting connected in between these two subsequent tyrosine amino acids? We are getting the 3. Now this thing that will be known as right now what? Triiodothyronine, or it's a T3. And here we will get also the T3. See here maybe we can get also T4. Here we will get what? T3. And in some persons we will get some amino acids. That means we can say some tyrosines. They will be still single. There will be no fusions. For example, in some cases. For example, over here we are getting on tyrosine is getting connected with on iodine, and they will still form a MIT. They will form the MIT, monoiodotyrosines. But yes, we can get also in this way that on more tyrosine molecule they are getting connected with only 2. There is no fusions. So this thing will be known as DIT. Okay. Now this type of the reactions, see, whenever we are getting the fusions of the 2 subsequent tyrosine molecules, at that time this type of the reaction is known as coupling reactions. So try to understand, on tyrosine molecule, for example, tyrosine plus iodide over here plus tyrosine, whenever, for example, the 2 iodide we are getting over here. This type of the reactions, whenever they will fuse with each other, this type of the reaction is known as coupling reactions. This type of the reaction that is known as coupling reactions. So what is the thing we got over here? First we got the iodide converted into iodine, then we got the transglobulin formations, then we got what? At first I said that what? Organification. Then I did talk about what? The coupling reactions. In the coupling reactions we'll get the fusions of the tyrosines amino acids with each other. Okay. Now we already did produce what? T3, T4, okay, all these things we got already over here. Like this we'll get many, for example, it's the thyroglobulin proteins. Over here we can get multiples T3, T4. For example, we can get the T3, T4 over here. We're getting the T3, we can get the T4 over here, we can get the T2, we can get T1. For example, we are getting over at T4, we are getting T3, we are getting T4, T3. For example, we are getting over here T2, T3, we are getting T4, T3, whatever. Okay, like this we'll get multiple. Like this we'll get what? The multiple thyroglobulin proteins that will have what? The product that will have that attaching. Along with that we'll get the attachment of the T3, T4, T2, T1 like this one. Okay. Now like this we'll get the productions of the what? T3 and T4. That means triiodothyronine and tetraiodothyronine will get in this way. Okay. Now we'll talk about these things. If we need, for example, we need. I'm erasing this part. Okay, guys. I'm erasing this part. For example, we need the T3 and T4. We need the T3 and T4 within the blood circulation. Okay, we need the T3 and T4 within the circulations. Now, how we get the T3 and T4 from here? That's the main thing. Now, let's, okay, 1 minute please. Let's check it on. Okay, no problem. So, I can continue. So, here we already got the T3, T4, T2, T1. Okay, along with that, what? The thyroglobulin proteins. Now, whenever we will need the T3 and T4 within the circulation, how we will get the T3 and T4 from here? So, basically we will get what? Basically we will get the productions of the T3 and T4. If we say in terms of the productions of the T3 and T4, definitely if the iodine intake is good, if the iodine intake is normal, at the time there is a more chances for the formation of diiodotyrosine. So, whenever there is a more chances for the formation of diiodotyrosine, at the time we will get the productions for the T4. Okay, that will be more. Whenever with the iodine intake that is normal or good. Okay, but for example, if we see the iodine intake that is not enough, or you can say in a small amount, at that time there is a chances for the productions of the more amount of monoiodotyrosines. As a result, we will get the chances for the formation of what? T3 more over here. Okay, and here we will get for the more productions of the more intaking of the iodide. Here we will get more chances for the productions of the what? Diiodotyrosines. Okay, there is a chances for the formation of diiodotyrosines. As a result, we will get the develop formations for the more chances of what? T4. Okay, now see, whenever we are talking in terms of the we need that the T3 and T4 in the circulation, at the time what will you do? At that time these whole molecules, for example, I am just showing these whole molecules in this way. Okay, now these whole molecules where we will get the T3, T4 and T2, T1. Okay, in this way. Now this whole part that will get fused within this membrane, and after fusing with this membrane by the process of pinocytosis, you can remember also, here we get the fusions over this one. Okay, and whenever they will get fused, they will get out the productions of the attachment of the T3, T4. And whenever they will get fused with this membrane, after that, by the process of pinocytosis or pinocytosis, these things that will be taken over within the, for example, we are taking these things over within the cells, and here we will get the attachment of the T3 and T4, T3, T4, T3, T4 like this. Now whenever it will be fused over here, okay, the lysozymes, within the, it says the lysosome is present, so lysosome that will be activated. And what we will see within the lysosomes, within the lysosome there are different kinds of the proteolytic enzymes. So for example, is the lysosome. Okay, the lysosome that will come and that will get fused with this what? With the thyroglobulin molecules, with the thyroglobulin protein, and as a result, what are the structures we will see over here? Okay, let me see over here. See, we'll get the attachment of the lysosome has along with these things, and we'll get over here whatever the you know, for example, so over here we got different kinds of the T3, different kinds of the what? T3 we can get the T4, here we can get the T2, here we can get the T1. Now here we will get the proteolytic enzymes with what? For example, if I show it over here, it will be more clear for you guys. For example, these are the structures, and here we got the attachment of the monoiodotyrosines. For example, I am writing it over here, MIT, or you can get the T3, we can get the T4, we can get the T2, we can get the T3, whatever. Okay. Now within the try to understand within the what? Within the lysosome there is the presence of the proteolytic enzymes. This proteolytic enzymes they will come over here and they will break down this whole protein molecules, and as a result we will get the productions of, in the free form we will get what? The T3, we will get also the T4, we will get also T2, we will also get T1. But what do we know? Within the blood we need only the T3 and T4. There is no use of T2 and T1 within the circulations. Okay. So from here we will get the T3 and T4 within the circulations, and whatever the T2 and T1, this T2 and T1, try to understand from here, there will get another enzymes. Okay, let's write it over here clearly. See, when you are talking in terms of the T2 and T1, we will get another enzymes that is present over here, that is known as thyroid deiodinase. Thyroid deiodinase. Let's write it over here. Thyroid deiodinase. Okay, so we'll get thyroid deiodinase. So they will just they will just got the they will got the detachments of the T2 and T1 from here, and they will be cycled back where? They will be cycled back again over here. They will be reused for the productions of the T3 and T4. Like this, we will get this type of thing. Now, if you show the structures over here, for example, okay, let me show this part. For example, if I say this is the molecules, protein molecules. Now see, if we get the attachment of the, try to understand, if we get the attachment, for example, the third and the fifth positions, iodine we are getting over here, and in this case we are getting on iodine. Now this thing that will be known as what? T3 definitely. Now for example, we are getting another molecule. In terms of the organifications, I am showing you guys. Okay, now try to understand. For example, we are getting the attachment of third and fifth positions. Same with third and fifth positions, we are attaching the iodine. This thing will be known as T4. Okay, now I am repeating once again these things. Okay, now please try to understand clearly. See, whenever I just talk about, we already got what? The thyroglobulin proteins, where we are getting the attachment of the T3, T4, T2 and T1. Whenever they will come within the thyrocyte cells, at that time whatever the thing we will see over here, we will see the fusions of the lysosomes. We will get the fusions of the lysosomes along with the what? Thyrocyte cells. Now see, along with the what? Along with these whole structures. That means whenever we will get the attachments of the lysosome, along with that what? Along with that thyroglobulin proteins. What? Whenever we are getting the attachments of the lysosomes, along with the thyroglobulin protein, where we will get the productions of the T3, T4, T2 and T1. After the fusions, we will get the within the lysosome there is a presence of proteolytic enzymes. These proteolytic enzymes that will be activated, and that will what? Break down this whole molecules, that whole protein structures in terms of the what? Thyroglobulin protein into, as a result we get what? The T3, T4, T2 and T1. So whenever we get the productions of the T3, T4, T2 and T1 for the using purpose within the circulations, we will take the T3 and T4. But as we don't need the T2 and T1, and there is a presence of another enzyme that will cause the recycling or reusing of the T2 and T1 again within the that will be sent over here,